So next we're going to go from the stomach down into the first part of the small intestine, which of course is the duodenum. And that takes us into the territory of the pancreas and the pancreatic juices. So pancreatic juice is the juice produced by the pancreas. And I think it's probably useful here just to have a little bit of background anatomy before we look at the details here. So the duodenum is going to start here just after the pyloric sphincter, and the duodenum actually curves around this way like this, and it goes up and carries on its way to where it joins onto the jejunum. So this would be the duodenum here. First part of the small intestine. Now the pancreas is actually located in this loop of the duodenum. So the pancreas is actually kind of located here like this. Pancreas is this kind of shape. And we know the pancreas is an organ with a head, a body and a tail. But the head is kind of nestled into this loop within the duodenum. Now there's a main pancreatic duct here, and this is going to collect pancreatic secretions from the exocrine tissue in the pancreas. And that's going to come down here. And there's actually a duct goes into the duodenum about here like this. But the main one carries on like this down there. That's the main duct there. Now this duct here, this superior duct, is called the accessory duct, and that's taking pancreatic juices into this more proximal part of the duodenum. But the main one is going through here, going into about 10 centimetres into the duodenum, so it's still the first half of the duodenum really, where the bulk of the pancreatic juices are going to be delivered into. And throughout the pancreas tissue there's clusters of exocrine tissue. Clusters of exocrine tissue, occupying about 99% of the bulk of the pancreas. And these are called acini, the acini, these clusters of exocrine tissue. And the cells in the acini are called acinar cells. So the pancreatic juices are actually produced by these acinar cells, going in and down the pancreatic duct here. But of course, just above here, we have another very significant organ, just here. Here we have the liver. This is where the liver is. And tucked underneath the liver just here we have the gallbladder, just there, with its cystic duct. And above that we have the common hepatic duct, and the right and the left hepatic duct tucked in behind the gallbladder. And then the common bile duct is going to go down here. So the bile is going to be produced in the liver, goes down the common hepatic duct, stored and concentrated in the gallbladder, then when required is going to go down the common bile duct. And the common bile duct actually goes through the tissues of the pancreas, and it joins up with the main pancreatic duct. So this last part of the duct is common between the pancreatic duct and the common bile duct. And this area where it's common is called the hepatopancreatic ampulla. Hepatopancreatic ampulla. In the old days it was called the ampulla of Vater. So here we just have bile. Here we just have pancreatic juices, but here we have both before being released into the lumen of the duodenum. Now, what is the other 1% of the pancreatic tissue made of? We've said it's 99% the acini, these clusters of exocrine tissue. Well, the other 1% distributed, about 1 million of them distributed all over the pancreas, are the well-known and loved pancreatic islets of Langerhans. And these of course produce the alpha cells in the pancreatic islets of Langerhans produce glucagon. The beta cells produce the insulin. But today we're thinking about the exocrine, acinar products of the pancreas. So let's think about what these products are. These products are in the pancreatic juice. So these are going to be in the pancreatic juice. And again, quite large volumes of pancreatic juice are produced. About 1200 to 1500 mils of pancreatic juice are produced every day. And we did comment when we thought about mechanical digestion that these volumes are important because they give the fluid medium. And the pancreatic juice will mix in the lumen with the intestinal juice, the succus entericus, to give us this watery environment. But the pancreatic juice itself, it contains water, of course, the enzymes, which we're going to look at in a minute, and also contains sodium bicarbonate. And this is important because this gives us the alkaline medium. The medium in the small bowel is alkaline, and the pH of the pancreatic juice is round about pH 8. So it's very much towards the alkaline side of the spectrum, altering the pH of the environment of the small bowel. Now again, because the pancreatic acinar cells are producing digestive enzymes, and as we'll see, these are going to be proteases, lipases and amylases, we don't want these protein digesting enzymes to be activated in the pancreas itself, because sometimes they are. So if a gallstone, for example, gets stuck in here, in the hepatopancreatic ampulla, then can you see that means that the digestive juices from the pancreas can't get out and they can dam back in the pancreas. Now we'll see that these are produced as proenzymes, inactive enzymes, but if there's an obstruction, then these enzymes can become activated, and that can mean that the pancreas will start digesting itself. This is a remarkably painful condition. This is the pathophysiological basis of pancreatitis. It's an auto-digestion of the pancreas. Or people that drink a lot of alcohol. The small ducts in the pancreas, especially people that are drinking alcohol over long time periods, the small ducts get blocked up. And again, if the small ducts are blocked up, the enzymes can't escape and we can get pancreatitis. So very painful condition of self-digesting of the pancreas. So it's absolutely vital that the enzymes are produced as these pre-enzymes or proenzymes. And one of the enzymes produced by the pancreas is trypsinogen. Trypsinogen. Now this is a protease, but it's a proenzyme. It's produced in inactive form, trypsinogen. Now, when it gets into the lumen of the small bowel, so once we're in the lumen of the small bowel, then of course we want this to start working. That's exactly where we want it to work. So we don't want it to work in the gland, but we do want it to work in the small bowel. So we need it to be activated once it gets into the lumen of the duodenum, which is protected by mucosa. And the trypsinogen is actually activated into trypsin by enterokinase. And enterokinase is what we call a brush border enzyme. It's produced by the enterocytes lining the lumen of the duodenum. So the trypsinogen will come into contact with the brush border of the duodenum. The brush border will contain enterokinase, and that will activate it into the active protease trypsin. So we now have activated trypsin in the lumen of the duodenum. Now, other inactive enzymes produced are chymotrypsinogen, and the trypsin, once it's produced, as well as digesting proteins on its own, will activate the chymotrypsinogen and convert that into chymotrypsin. So trypsinogen is first activated into trypsin by the enterokinase, then the chymotrypsinogen is activated into chymotrypsin by the trypsin. And the trypsin will also activate these as well. This is Procarboxypeptidase, another protein digesting enzyme. It will digest that, it will activate that rather, into carboxypeptidase. Again, trypsin will do this. And it's the same with the protease, the proelastase will be activated into elastase. And the elastase is going to digest elastic tissues in the food that is consumed. So these enzymes here are all active proteases. Now, you might wonder why we've got 4 different protein digesting enzymes. Well, the answer is that these different enzymes will actually work on different peptide bonds. So you remember in our proteins we have these different long chains of amino acids, different peptide bonds. So different protease enzymes will break different peptide bonds between pairs of amino acids. That's why we have these different variety of protein digesting enzymes. Now we also notice that the pancreas produces pancreatic amylase, and the amylase is going to break down carbohydrates into simpler sugars. And the pancreatic amylase, as well as breaking down starch, which is sometimes called, well, starch is vegetable starch, it also breaks down glycogen. The glycogen is this huge polysaccharide molecule of usually glucose units. Well, it is glucose units, but it comes from animals. So starch comes from plants, glycogen comes from animals. In fact, glycogen is sometimes called animal starch. So that will break down those carbohydrates. But there are some polysaccharides, there are some carbohydrates that amylase will not digest. So which polysaccharide carbohydrates will pancreatic amylase not break down? Well, the answer to that is cellulose containing polysaccharides. And in fact, anything that's not broken down by the gastrointestinal tract, such as cellulose, is a fibre, dietary fibre. So dietary fibre is not acted on by any of the digestive enzymes, and that means chemically it goes out as it came in. And of course we know that water-soluble fibre and non-water-soluble fibre are very important for health and for the health of the gastrointestinal tract. But the reason that they're fibres is because they're not acted on by digestive enzymes such as amylase. Pancreatic lipase, of course, is going to start breaking down or continue the breakdown of fats. Now, most fats, when they're consumed, they contain a glycerol unit, and they contain 3 fatty acids because they're triglycerides. So this will be the glycerol unit, and these will be the fatty acid units in the triglyceride. So the pancreatic lipase is going to break down these triglyceride fats. It breaks them down into monoglycerides, which is a glycerol with 1 fatty acid left, and the other 2 fatty acids are going to be liberated. So we end up with monoglycerides and fatty acids, and the fatty acids can be long chain or short chain fatty acids. So we could carry on illustrating this graphically. We're going to end up with monoglycerides and 3 fatty acid units, 3 fatty acid chains, long chain or short chain. So that's what the pancreatic lipase is going to do, breaking up the triglycerides into glycerol, monoglycerol and monoglycerides and fatty acids. Now, in the food we eat, of course, is going to contain ribonucleic acid, RNA, in the cells, and also deoxyribonucleic acid, DNA, in the nuclei. And we notice that the pancreas produces specific enzymes for those. So there's a ribonuclease and a deoxyribonuclease, breaking down RNA and DNA. Now, most of the products of digestion are going to be absorbed through the jejunum and through the ileum. And we've looked before at this very large, at least 250 square meters of internal surface area through which absorption takes place. But as well as just absorption, there's going to be a final stage of digestion. And this occurs in what we call the brush border enzymes. Now, what's happening here is the cells actually lining the mucosa, as we've noticed, are these columnar cells, column-shaped cells like this. We noted on previous videos they have very tight junctions that interlace with each other. So we've got these enterocytes, and on the surface of the enterocytes we have these microvilli, and we have noticed that the microvilli are clearly discerned with an electron microscope, but prior to that this looked, with light microscopes, this just looked like a brush border because of all the little hairs in it. So great profusion of these microvilli from the surface of the enterocytes. And here are the food products of digestion in the lumen of the small intestine, and of course they need to be absorbed through these cells into the submucosa, where the rest of the mucosa, and the submucosa where the blood and lymphatics are, in the villi, for example. Now, the phospholipid bilayer membranes on the surface of these contain digestive enzymes, different digestive enzymes. Different digestive enzymes. So that means when the products of digestion are absorbed, they've got to go through this membrane, and this membrane contains these enzymes, and as they go through this brush border membrane, the digestive process is going to be completed. So these are what we mean by the brush border enzymes, literally in the phospholipid bilayers of these brush border microvilli. And what happens here is there's some enzymes, so we can see there's a sucrase, lactase, maltase, aminopeptidase, dipeptidase. These are the enzymes, or the main enzymes in the brush border. And what are they doing? Well, the sucrase in the brush border is finishing off the digestion of the disaccharide sucrose, converting it into the monosaccharides glucose and fructose. The lactase is digesting the lactose, which is the milk sugar from milk, and it's digesting that into the monosaccharides glucose and the monosaccharide galactose. Maltase is taking the products of starch digestion, maltose, which is a disaccharide. There's also some 3 sugar units here as well, but the maltose is a disaccharide, and that's being converted into a molecule of glucose and a molecule of glucose. And then these last 2 are proteases, protein digesting enzymes. So there's aminopeptidase, which cleaves amino acids at the end of a peptide, and then the final one, dipeptidase, splits. Dipeptidase, as the name suggests, when there's 2 amino acids still linked together by a peptide bond, it splits that final peptide bond to give single amino acid units. So all we've got going in here, by the time we get through this brush border membrane, past these, when the products of digestion have run this gauntlet of brush border enzymes membrane-bound enzymes. All we have in here is single amino acids and single monosaccharide sugar units. And there's also active processes of absorption facilitating the absorption of these things. And fatty acids and monoglycerides. Now the fatty acids and monoglycerides, they can actually diffuse into these cells just by simple diffusion, because they're fatty and the membrane is fatty. So these just go into the cells. The amino acids and the monosaccharides, the sugars and the protein fragments, they need to be absorbed via active transport, but the fatty material can just diffuse straight into the cell. So what we get is the fatty acids and the monoglycerides. They go into these enterocytes. So these are the enterocytes here. And here they're converted into balls called chylomicrons, which then go on and go into the lymphatics because they're too big to go into the blood capillaries. So the chylomicrons are produced in the enterocytes and go into the lymph. But as far as I know, there's no final fatty digestion takes place as it goes through the enterocytes, as there certainly is for the carbohydrates and the products of carbohydrates and the products of protein digestion. So we started off with large insoluble molecules, and what we have is simple sugars, monosaccharide units, single amino acids going into the blood, and we have chylomicrons containing fatty acids and monoglycerides going into the lymphatics for removal to the circulatory system. The digestive process is completed even as the final products are being absorbed by these fascinating cells, the enterocytes.